Okay, good afternoon. So today we're going to start with another system that is so closely related to the cardiovascular system. It is the lymphatic system. These are the components of the lymphatic system. There are organs, lymphatic organs. We'll talk about them in a moment. There are primary and secondary. And there are lymphatic vessels, which start at capillaries, then lymphatic vessels, then trunks, and then ducts. And there is the lymph fluid, which is usually it's a clear fluid. Most of it is a clear fluid, and it's the same as the interstitial fluid, the fluid that is present in the extracellular compartment, as well as the bone marrow, because in the bone marrow, the cells of the lymphoid system, which also belong to the blood, they are produced in the bone marrow, and some of them even they mature in the bone marrow. So that's why the bone marrow is also considered as part of the lymphatic system. Lymphatic organs include some of them you are familiar with, like the thymus here in the thorax, the spleen, which is located in the abdomen, and then we have multiple groups of lymph nodes and lymphatic nodules. The lymphatic nodules are different from lymph nodes in that they are present in the wall of certain structures. Like, for example, they are present at the beginning of the respiratory and digestive passages, like the tonsils, and they are not covered by connective tissue. They look like lymph nodes. The name indicates as if they are lymph nodes, but they are nodules. A lymph node usually has a capsule, and it receives an afferent and efferent lymphatic vessel, as we will discuss in a moment. So there is a difference, in other words, between a lymph node and a lymphatic nodule. The function of the lymphatic system is that it drains the excess interstitial fluid. Not all the fluid, the interstitial fluid, is taken up by the capillary bed. You can see it here in this diagram, that the cardiovascular system will bring the plasma to the tissue, and through, at the end, there are capillaries. This is the arterial end of the capillary, where nutrients and oxygen go to the extracellular fluid, and then the metabolites, carbon dioxide, will return back by the veins. Some of the constituents of this extracellular fluid will not be able to return back, either lagging or because they have large size. Like sometimes there are leaking proteins, let's say, and they cannot return. They are bigger in size. Sometimes there are lipids, like the ones that are absorbed in the small intestine, so they cannot return by the cardiovascular system, so they will return by these green capillaries. These are the lymphatic capillaries, and you can see that the lymphatic capillaries, they start as blind-ended. There's no circulation. They start as blind-ended because eventually they are going to drain into the blood circulation. So they don't have two ends. And then the fluid also transports dietary lipids, as I mentioned, because they are large molecules, cannot be transported from the gastrointestinal tract, and they facilitate immune reactions, as we will see in the lymph node. When there is infection, the lymph nodes enlarge, for example, because the lymphoid tissue proliferates and facilitates the immune reaction by either producing antibodies or by killing the invading organisms and phagocytizing them. The lymphatic vessels, as I said, they start as capillaries. You can see the capillary here. It's a blind end capillary, and then these capillaries become larger and larger vessels. They form trunks and ducts, and they open here in the venous circulation. They open either on the right or on the left. So they open either on the right or on the left. The same place. It is the place where the large vein that drains the head and neck, which is called the internal jugular vein, meets the vein that drains the upper limb, the subclavian vein. At this angle, which is called the venous angle, so two lymphatic ducts open. On the left side, it is the thoracic duct, and on the right side, it is they call it the right lymph duct. Right lymph duct, easy. The other one is called the thoracic duct. I'll talk about them in a moment. The lymphatic vessels, they have a structure that looks like a vein, but they are even thinner than a vein, the wall. Remember the wall of the vein that we were talking about last time? And they have valves as well. So even the same mechanism of pushing the lymph back to the circulation is applies for the lymphatic vessels. So like respiratory movements, valves, surrounding muscles, all of them, they will push the lymph back to the circulation. This is again more focusing on the beginning of the lymphatic capillaries. As I said, that they are blind-ended. They are lined by endothelial cells like the blood vessels. And in addition to that, which is not present in the capillaries, is that they have what we call anchoring filaments. Look at these anchoring filaments. These anchoring filaments, they pass in between the surrounding cells, the tissue cells, okay? So they will prevent the collapse of the capillary. If there is increase in pressure here in the extracellular compartment, then these will be, the increase in pressure will anchor these filaments and prevent the collapse of the capillary, because these filaments are anchored to the tissue, so the endothelial cells will not come close together. At the same time, the other difference between the blood capillary, in addition to the presence of anchoring filaments, the other difference in the lymphatic capillary is that, as you can see, that the endothelial cells are overlapping. Okay, so they overlap each other. So when there is increase in pressure, one of them will be pushed, and this will create a larger gap in between the cells. It will not cause collapse, but at least push one of the ends and will cause a larger gap between the cells. That's why larger molecules can gain access to the lymphatic vessels, lymphatic capillaries, but not to the blood capillaries. So these two mechanisms are clearly shown in this diagram. Then we have the lymphatic vessels that are present, or capillaries that are present in the intestinal villi. We're going to talk about the gastrointestinal system, and you will find that there is a central lymphatic vessel in each of these finger-like projections called the Lacteal. And these are specifically for removing the fat and fat-soluble vitamins that are absorbed from the gastrointestinal tract and cannot gain access to the capillary circulation. And that's why the lymph that is present in these vessels, in specific, it is milky in color because it contains a lot of lipids, and it is called chyle. It's not yellow, it's not transparent like the rest of the lymph. It is chyle, and they are called the Lacteals because they contain fluid that looks like milk. Lact means milk. So as I said, that on the right venous angle, the angle between internal jugular vein on the right side and the subclavian vein on the right side, opens a very short right lymph duct. And this brings lymph from about 1/4 of the body on the right side above the diaphragm. So the right chest, right upper limb, and right side of the head and neck. While the remaining 3/4 of the body, they are drained by, on the left side, at the venous angle on the left side, by what we call the thoracic duct. So we have the left head and neck, left side of the thorax, left upper limb, and the entire, both sides of the abdomen, both sides of the pelvis, both lower limbs, everything below the diaphragm is drained there into the thoracic duct. If we go back a little bit here, we will see that the thoracic duct starts in the upper abdomen as an elongated sac called the cisterna chyli. You need to memorize these names, unfortunately. So cisterna, cistern means like a sac, chyli because it contains chyle, the chyle that was brought from the intestine. So that's why it's called the cisterna chyli, and it's about like 2 inches in length, and at the apex of from its upper part starts the thoracic duct. It's called thoracic duct because it enters the thorax, and then the thoracic duct deviates to the left as you can see, and opens into the left venous angle. So that's why everything below the diaphragm on both sides will go to the thoracic duct. But on the right side, the right quarter of the body above the diaphragm goes to the right lymph duct. As I mentioned, that fluid and protein escaping from the vascular capillaries will go back to the lymphatic capillaries, and there are valves, there are respiratory movements, muscular contraction, even the pulsations of the nearby arteries will be helpful. You know, the arteries are pulsatile, so this, and because the wall of the lymphatic vessels, as I mentioned, are very thin. They are even thinner than the walls of the veins. So that's why any pressure outside can affect the contents of them. When there is excess, when these lymphatics fail, there's excess, not necessarily they fail, but there is excess extracellular fluid that cannot be taken by the lymphatics. We will have a condition which is called edema, as you can see it here. This is called pitting edema. There is excess fluid in the interstitial space, either because one of the reasons is this happens, for example, in liver diseases. So the liver will not be able to form proteins. So that's why the level of plasma proteins will be less, and this will reduce the osmotic pressure of the blood. So the blood cannot return, the extracellular fluid will not be able to return as efficiently to the venous part of the circulation. Or sometimes, like, there is loss of proteins in, for example, kidney diseases. Or sometimes there is heart failure. Heart failure on the right side of the heart. If the right side of the heart fails, cannot pump the blood, then the blood will be collected back and back, especially in the lower part of the body, venous blood, and so the venous pressure will become higher because the heart is inefficient to pump the blood from the right side to the lung and then return it back to the left side of the heart. So these are causes of in general, some of the causes of the edema. As I mentioned, that the lymphatic system also promotes the immune reactions, and it has cells of the immune system. These cells are mainly, they are the lymphocytes. They are the main defense cells. The other cells are neutrophils. They are the white blood cells in general, macrophages, which are derived from monocytes. But the lymphocytes are the main defense cells of the immune system. And we have B-lymphocytes and T-lymphocytes, as we mentioned previously, that the B-lymphocytes are produced in the bone marrow and they mature in the bone marrow. The T-lymphocytes are produced in the bone marrow but need to travel to the thymus, where they become mature by the effects of the hormones that are produced in the thymus. And you cannot differentiate between them structurally. So the B lymphocytes, when there is a trigger, when there is an immune reaction, they will differentiate to form plasma cells, as you can see them here. And the plasma cells will produce proteins called immunoglobulins or antibodies. And these antibodies will attach themselves to, for example, the bacteria invading organism. And so this organism will become easier to become phagocytized, to be identified and recognized by other cells of the immune system to destroy it. So these are usually, this is a short-lived reaction, but some of the B lymphocytes, they do not form plasma cells. At the same time, they form other types of B lymphocytes, which are called memory cells. And these will remain circulating in the blood, so that if the same organism is going to invade the body, then there are cells that will immediately remember and produce antibodies. It doesn't require an extra time to be recognized. These memory cells could be either T cells or B cells, and they are the basis of vaccination. That's why, for example, when you vaccinate, then it means that you are inducing the formation, you are probably introducing the microorganism, either living, attenuated, or dead, and this will induce the immune response to produce a small local reaction, but at the same time, it will induce the formation of memory cells that continue circulating in the blood and can hit that organism if it invades the body the second time. The T lymphocytes, they do not produce antibodies. They do not proliferate into plasma cells and produce antibodies, but there are different types of them. One type of them is the main type, is the killer cells or the cytotoxic T cells. They inject cytotoxic. Cyto means cell, toxic, killing. So they inject substances that kill and destroy either the tumor cell or the invading organism. And there are other types of these T lymphocytes. Not all of them are cytotoxic. You will study that probably later on in the microbiology or even in the pathology. There are helper cells, which they increase the activity or amplify the activity of the immune reaction. There are regulatory suppressor cells, which will stop the immune reaction. As when the invading organism is destroyed, there is no more organism, then the reaction should stop. Otherwise, these killing cells are going to destroy the body itself. And this is what we call autoimmune reaction. And there are memory cells. So these cells are functional types. They are not structural types. You cannot differentiate between them structurally. I'm talking about them as functional types of T lymphocytes. The organs of the lymphatic system, we have primary and secondary lymphatic organs. And the primary lymphatic organs, the cells of the lymphatic system are produced and mature. And therefore, we have the bone marrow and the thymus, because they cannot mature elsewhere. In the secondary lymphatic organs, it is where the immune response actually takes place. So, like for example, lymph nodes, lymphoid follicles or nodules, the spleen, because there are no lymphocytes maturing or produced in the spleen or even in the lymph nodes. So that's why they are called secondary lymphatic organs. You can see the distribution of the lymphatic organs, the primary and secondary. So the tonsil, lymph nodes, these are secondary. Spleen, Peyer's patches, they are lymphoid follicles like the tonsils, but they are present in the wall of the small intestine, especially in the terminal part of the ileum. So the lymphatic system could be diffuse. You can see in the submucosa, let's say, if you have a section of the submucosa of the trachea or of the esophagus, you will find that there are diffuse lymphocytes in the submucosa. But in certain places, these lymphatic cells will aggregate together and form like these nodules. This is 1, 2, 3, 4, 5, 6. So you can see multiple lymphatic nodules around a lumen. Actually, this is a section in the vermiform appendix. And this is the function of the appendix. It is one of the places where the immune reaction can take place because its wall contains a lot of aggregates of lymphoid tissue in the submucosa and in the mucosa. This is what they call mucosa-associated lymphoid tissue. They call it MALT. Okay, and that's why if there's any like infection, then these lymphoid nodules, they are going to proliferate, they are going to enlarge, and when they enlarge, they will obstruct the lumen. And if the lumen is obstructed, then the appendix has to be removed. Peyer's patches are located in the terminal ileum, as I mentioned. The tonsils, we have met some of these tonsils when we studied the respiratory system. I mentioned that some of them are present in the nasopharynx, called adenoids. Some of them are present in the oropharynx, which you can see. These are the tonsils or the palatine tonsils. There are tonsils at the posterior third of the tongue that produce the cobblestone appearance of the mucosa of the posterior third of the tongue. So they are present in a strategic place at the beginning of the respiratory and digestive passages. These are not lymph nodes, they are lymphoid tissue because they are not surrounded by a connective tissue capsule. What is surrounded by a connective tissue capsule is a lymph node. Generally, the lymph nodes are bean-shaped. Some of them are so small and tiny. Some of them are about 1 inch in length, like the ones that you see here in the inguinal region, or even the ones that are present in the abdomen. They are quite a big size, and they are located along the lymphatic vessels. So that's why they filter the lymph. So for example, let's say here there's a pinprick infection or any infection in a small wound on your finger, then the infecting organisms will pass through the lymph. They gain access into the lymph because they are large size, and they will pass up until they reach the first group of filters. Here is in the axilla, and so in the axilla they will start inducing the immune response, and the cells will proliferate, and so the axillary lymph nodes increase in size. That's why you will be able to feel them, because they become larger in size, they become tender, hot, because of they are the first filter. Okay, same thing is true if there is an infection in the sole of the foot, let's say, for example, then the first filter will be here in the inguinal lymph nodes, and then the second filter and third filter, and so on. This is the same thing. This is the same thing for a tumor. Like, for example, in the breast, as you can see, the breast mainly drains into the axillary lymph nodes, but some of the medial part of the breast drains into lymph nodes that are present in the thorax. So that's why whenever there is a suspicion of a tumor in the breast, then the first thing to examine are the axillary lymph nodes. And there are certain features, clinical features, to give an indication whether the enlarged lymph node is cancerous or it is infected. So as you can see here, cancerous lymph nodes, they are non-tender, they are fixed, while the infected nodes, they are a little bit softer and they are tender, and they are movable. So the lymph nodes, they filter the lymph, and not all the lymph nodes are palpable because some of them are deep. So for example, you have lymph nodes in the abdomen and pelvis. You cannot palpate them. There are lymph nodes in the thorax. Even in the cervical region, there are deep group of cervical lymph nodes. But nevertheless, there is a superficial group of cervical lymph nodes which you can feel. The axillary lymph nodes you can feel. The lymph nodes in the groin, in the femoral triangle, around the femoral artery and the femoral vein, again you can feel these lymph nodes, and that's how a clinical examination is done. A lymph node consists of a capsule. As I said, the essential thing is that they have connective tissue capsule, and from the capsule of dense irregular connective tissue, there are septa, walls, trabeculae, okay, that divide the lymph node into compartments, and then there is a framework of reticular fibers. Remember the fibers of connective tissue that we dealt with at the beginning? There are collagen fibers, there are elastic fibers, and there are those tiny reticular fibers. So those are the reticular fibers. They provide a scaffold for the lymphatic cells, support them. And there is a region here which is called the cortex, and there's another region which is called medulla. The essential thing that you notice is that each lymph node has an afferent vessel or multiple vessels and efferent lymphatic vessels. So the lymph will come from another place, filtered here through the cortex and then medulla, and then leaves the lymph node out to reach another lymph node or to reach another lymphatic vessel. In the cortex, usually there are B-lymphocytes which produce plasma cells, and when they proliferate and become active and produce plasma cells, the center of the follicle here will become lighter in color. We call it a germinal center. So when it becomes very active, then the center will be lighter in color, called germinal center. And then we have the medulla. The spleen is the largest single mass of lymphoid tissue. and this is the location of the spleen. We will mention the spleen several times when we deal with the GI tract because it is located in the left hypochondrium below the diaphragm, as you can see here. It has a hilum. Remember the word hilum, the term hilum, where structures pass in and out. So here, the hilum, we have splenic artery, splenic vein, and lymphatic vessels going into the spleen. So fragile, the spleen. Although it contains a lot of blood and it has a fragile tissue. And again, it has a capsule of dense connective tissue with multiple septa, but there is no cortex and medulla. Cortex and medulla is present in the lymph node, but there is no cortex and medulla here. But we will have aggregates of lymphoid cells surrounding a blood vessel, and this is what we call the white pulp. It doesn't look, it's not white in color, but because these cells are white blood cells originally, so that's why it's called white pulp. And then there are other areas here which is called red pulp because it is filled with blood. So here what happens is that the lymph reaches the spleen. There is no afferent and efferent. There is only afferent lymphatic vessel, as well as the blood reaches the spleen. So if there is if the infection has reached the blood, then there is a way that the blood is going to be filtered in the spleen, and the invading organisms are going to be attacked by the lymphoid tissue that is present in the spleen. So if there is a microbe here in the central artery, it will filter itself in the white pulp around it and will be attacked by the lymphocytes. Either B-lymphocytes that are going to produce the antibody, or the T-lymphocytes that are the killer cells that are going to inject cytotoxic substances into it. So that is the importance of the white pulp. Why do we have the red pulp then? The red pulp, which is mainly formed of blood cells, RBCs, mainly RBCs. There are capillaries that will bring the blood to the red pulp, and then the blood will return back via venules, as you can see here, large venous sinuses. While doing so, the red blood cells that are defective, the defective red blood cells are going to be attacked by the macrophages and removed from the blood. That's why we mentioned earlier that the red blood cell has a lifespan of 120 days. It cannot repair itself. So when worn out RBCs, defective RBCs cannot be repaired, and so they are going to be destroyed either in the liver or in the spleen. And this is in the red pulp of the spleen. This is the other function of the spleen. Here you can see that the spleen is closely related to the lower ribs, so that's why fractures of the lower ribs on the left side might result in injury of the spleen, rupture of the spleen. It's so fragile. Sometimes it's an indirect trauma, like for example, a car accident and a hit on the abdomen by the steering wheel will cause rupture of the spleen, and because of its profuse blood supply, it will cause a very dangerous internal bleeding. And once it is injured, then it cannot be sutured because it's so fragile, and that's why it has to be removed, splenectomy, and its function will be taken over either by the bone marrow or by the liver. The thymus, we mentioned about the thymus. It's located in the mediastinum. It is larger in infants and most active in childhood, but then after that it regresses, does not disappear, its function does not stop, but it regresses, and most of it will be replaced by fatty tissue. So when you dissect, you will find like only remnants of the thymus and surrounded by fatty tissue. This is how the thymus look under the microscope. Again, it has a capsule and trabeculae of connective tissue. Divide the thymus into lobules, and there is a cortex here. This is the cortex, and a medulla. The medulla is lighter in color, and it is in the cortex that the maturation of T lymphocytes takes place because of the hormones that are produced by cells that are present in the thymus, by thymic cells. And then at the same time, there are macrophages. So if any of the T cells fail to mature, or if they die, then they are going to be attacked by these macrophages and dumped into the medulla. So you will have these structures, which are a characteristic feature of the thymus, called Hassel's corpuscles, thymic corpuscles. They consist of concentric layers, concentric, like this. Okay? So concentric layers of flattened dead cells, okay, called Hassel's corpuscles. They are present in the medulla of the thymus. The mature cells, the cells that became mature in the cortex, also will reach the medulla, and then they leave the medulla through the blood and reach the other tissues.